The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and thank you for joining us today. You have logged on to the Nelson Labs webinar, the Reprocessing Rethought, Approaches for Cleaning Validations of Single-Use 3D Printed Devices Processed at Healthcare Facilities. We'd just like to take a few moments to do some housekeeping items. If you would miss one of our webinars, or would like to refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can also receive notifications for upcoming live and on-demand webinars by following Nelson on Facebook and on LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will also find curated content from our industry thought leaders from across the Sotera Health brands of Nelson Labs, Sterigenics, and Nordion. Today we are joined by Brianna Barber. Brianna has worked at Nelson Labs for over 6 years, specializing in cleaning and disinfection validations for reusable medical devices. She has had experience with hands-on testing, directing studies, regulatory affairs, and consulting. In her current role as Consulting Study Director, she is the primary contact for sharing expertise related to testing requirements and ensuring medical device manufacturers are set up for successful regulatory submissions. Brianna graduated from Arizona State University with a master's degree in regulatory science in 2023. She presents at seminars throughout the United States and participates in review of standards, and is passionate about sharing her expertise on regulations for reusable medical devices and supporting customer needs. We do encourage your questions today, so go ahead and submit them in the questions box at any time so that we can have some discussion after Brianna's presentation. Now, beginning today's presentation, I'd like to turn the time over to Brianna.
Thank you, Mike. Hello, everybody. I appreciate you joining on this lovely Tuesday, and we are closer to the weekend, but not quite. It's better than Monday, at least. So today I want to talk about approaches for cleaning validations of single-use 3D printed devices. And the reason for this is due to the increase we have seen in the popularity of 3D printed devices. We see them come through quite a bit more. They're becoming more unpopular due to the lower cost and ease of manufacturing. And I wanted to bring attention to some challenges and gray areas for regulatory reasons. And yeah, here we go. Many of you likely already know what a single-use device is, but a single-use medical device is defined as a device intended for one use on a single patient for a single procedure. Now, a 3D printed medical device is a device created using additive manufacturing and building three-dimensional objects from digital designs. As you know, 3D printed devices could be an abundance of different materials. And sometimes these materials can present unique challenges to cleaning and/or disinfection and sterilization. But today we're really going to focus on the cleaning aspect. Now, something to consider is that for these types of devices, they are within scope of certain guidance and standards that might not be known. As we get into this presentation, I want to just go over the flow of the process of these types of devices. So we start with the manufacturing of the device. It is then sent to the healthcare facility. It is processed at the healthcare facility through cleaning and disinfection or sterilization, and then it is ready for patient use. And we're really focusing on this processing at healthcare facilities portion. Now, these standards, AAMI ST98 is the standard for cleaning validations of healthcare products. And then the FDA guidance document is a guidance document for reprocessing medical devices in healthcare settings. In both of these documents, single-use devices are included in the scope. As you can see, for AAMI ST98, it applies to all medical devices that require cleaning prior to each clinical use of that device. For the FDA guidance document, it applies to not only reusable devices, but single-use devices initially supplied as non-sterile to the user that require processing before use on the patient. Now, there are other standards and guidance out there for single-use devices, but these are the two that I really wanted to bring awareness to, as they might not be known that these single-use devices are inclusive. And there is a difference between these types of cleaning validations versus cleaning validations for manufacturing residuals. And we're going to focus not necessarily on the manufacturing residuals of 3D printed devices, but aligning with the standard and guidance for these types of cleaning validations, evaluating the effectiveness of the cleaning. So a typical cleaning validation really has three main points to it. There, of course, is a lot that goes into these types of validations, but I'm going to focus on kind of the three major areas. We have soiling, cleaning, and endpoints or analytes. When it comes to soiling, the standard approach for soiling a device is to mimic clinical use. So what that means is you select a test soil, so a contaminant, that mimics the use of the device. As an example, a reusable device such as an orthopedic surgical instrument, we would select a test soil that may have some blood in there and some bone, mimicking what it would see during clinical use. And then we would use a soiling method such as maybe handling the device with our gloved hands that were soiled with the test soil, or maybe immersing it into the soil. Now, I will get to the challenges for 3D printed devices, but just keep in mind that clinical relevance. That is going to be one of the biggest challenges and gray areas. In terms of cleaning, typically for a cleaning process, we want to select a detergent that is based on the market you intend to submit your device to. So in the US, neutral enzymatic detergents are preferred, whereas over in the EU, alkaline detergents are preferred. But that isn't the only thing you want to consider. You also want to consider compatibility of the detergent with your device. And typically with cleaning instructions, we try to develop simple yet effective instructions. When you take a look back or a look into human factors perspective, if you have a 2-hour manual cleaning process, that is going to be difficult for the end users in the sterile processing department. So it's important to consider that when we're discussing selecting cleaning processes. And then for endpoint selection, the most common endpoints we see for validations are protein, total organic carbon, and if the device is patient contacting, cytotoxicity. And then I put manufacturing residuals here as well, even though that is a separate discussion. For 3D printed devices, keep in mind you will need to evaluate manufacturing residuals, such as particulates, THC, TOC, total organic carbon, is also something you could do for a manufacturing residual. But these are kind of just the basic approaches we would take for a normal cleaning validation per ST98 and the FDA guidance document for reprocessing. Now, when we get into 3D printed device validations, I know the term gray area is like, oh my goodness, but it really is a true issue. You know, with these two documents, ST98 and the FDA guidance document, they seem to be more geared towards reusable devices, even though they still apply to those single-use devices that require cleaning at the facility prior to use. So when it comes to soiling, something to consider is the material of your 3D printed device. In my experience with some materials, they like to soak in the test soil. So we need to consider what kind of test soil do we use. And the other challenge is clinical relevance. For these devices, we're not necessarily going to be cleaning post-use. This cleaning validation is performed prior to the initial use. So when we are soiling, is it appropriate to follow the standard and guidance document and mimic clinical use? Or should we consider maybe just mimicking contamination the device might see prior to the cleaning and processing procedure, such as being handled with bare hands or sitting in the sterile processing department, potentially near other dirty devices? There's a lot of different things to consider when it comes to soiling. So those are just some thoughts to keep in mind. And when you go to select a test soil, just consider maybe we shouldn't be using a blood soil, even if that device is going to contact blood, because that's not really relevant to the purpose of the cleaning validation. And then when we move into cleaning, something to consider is the material and compatibility with the detergent chemical composition. So something that might be beneficial would be to contact the hospital or reprocessing location that will be processing your 3D printed device and see what detergents they use and look at those detergents and see, oh, is my material compatible with this composition? And just keep in mind, yes, it's only going through this one initial cleaning, but you don't want to cause any degradation or damage that could impact the device and potentially impact the patient from this single cleaning process. And when it comes to endpoint selection, one thing to consider too is materials. Are your materials going to interfere with certain endpoints? For example, total organic carbon is a pretty sensitive assay, so just keep in mind that that could also have an impact. And clinical relevance. To align with ST98 and the FDA guidance, you should really select endpoints that are clinically relevant. But in this case, I think for 3D printed devices, you know, looking at protein and looking at total organic carbon and looking at the cytotoxicity post-cleaning are good endpoints to select, especially because protein is kind of a catch-all endpoint. Total organic carbon is great because it is also used for manufacturing residuals, so it can be kind of a win-win situation. You can knock out one of your manufacturing residual tests and kind of combine them into this test plan for these types of validations. And with cytotoxicity, if your device is patient contacting, it would be critical to assess the detergent residuals, and cytotoxicity is just one method that does have an acceptance criteria associated with it. But there are other methods out there as well to assess detergent residuals, but it is something you'll want to do. You know, you're taking your device through this cleaning process and then it gets used on a patient. And we want to ensure that there's nothing harmful remaining on the device from the cleaning that could impact the patient. So these are really just some challenges or kind of a gray area that should be considered. You know, how do we soil to align with these standards, but also get the best data out of these types of validations, selecting cleaning detergents and methods and endpoints. And that is it for my presentation today. So I will open up the floor to any questions.
Thank you, Brianna, for all those great insights and everything that you've shared today with all the information that you have. You've given us a lot to think about. We do have some questions, and we'll just dive into those. So here's a question. It says, why are healthcare facilities cleaning a single-use disposable device?
Yeah, so there are some cases where a manufacturer of a 3D printed device is not going to have the healthcare facility clean it. So for example, the cleaning occurs post-manufacturing at the manufacturing site, and then it gets packaged and sent to be sterilized at the healthcare facility, or maybe it goes through a sterilization prior to arriving. But there are cases where there are certain manufacturers that will require a cleaning per IFU, or instructions for use, followed by disinfection and or sterilization prior to use, even for single-use devices. So it really is dependent on the manufacturer of the device.
Great, thank you. And if anyone has questions that are here attending, please go ahead and send those in through the questions tab on your on this meeting. Our second question here, Brianna, says, what would the recommended sterilization procedure be for these types of devices? I'm assuming for reusable devices.
Yeah, so for these types of 3D printed devices, really it will be material dependent. You want to ensure that the sterilization process is not going to damage the device. So, you know, if your materials allow for steam sterilization at those higher temperatures, like 132 degrees Celsius or 134 degrees Celsius, and those kind of classic US and EU steam cycles, then that would be a good option. If your device cannot really withstand that high temperature without the risk of damaging the materials, you could do, a different method such as EO or vaporized hydrogen peroxide, but those can get a little tricky because you might have to consider residual testing for those sterilization methods.
Okay, great. We have lots of questions coming in. Here's another one. Are you aware of any particular 3D materials that do not hold up to cleaning methods?
As far as I'm aware, I don't have much experience with materials not holding up to cleaning processes. It's more cleaning detergent composition. Some materials might not be able to handle, an alkaline chemical composition, while others might be okay. It's really material dependent and detergent dependent. So I would be more than happy to have a discussion after this presentation on your device materials and what cleaning method or detergent you plan to use, and we can go from there.
Okay, great. Here's our next question. If the hospital prints the 3D device on site, would it still need to be processed prior to use?
I would say yes. If once the hospital prints the device, there should be some sort of cleaning and or disinfection and sterilization method in place prior to using it on the patient. One, to remove, any manufacturing residuals or any residuals like that. And two, just for the safety of the patient, to ensure that they have a clean and disinfected and or sterile device.
Perfect. Here's another one. TOC testing, is that a part of extractables and leachables testing or a separate test? I'm assuming maybe it's not asking a question around is separate from cleaning validation.
Yeah, so the total organic carbon testing, TOC testing, is a part of the cleaning validation. So it is something where we, post-cleaning, will extract the device in a water extract, and then here at Nelson, at least, we transfer to our chemistry department and they run the standard TOC assay. And any further kind of technical discussion about total organic carbon, I would definitely refer you to one of our chemistry experts here at Nelson Labs.
Okay, great. A few more questions here. To confirm, the endpoint selections are options, but not all are required depending on the product usage. They're asking if that's correct.
Yeah, that is correct. It is dependent on the criticality of your device as well as relevance to your device. So for example, a non-critical 3D printed device, so something that either does not contact the patient or would contact the patient but only intact skin, typically we would recommend testing one endpoint, and that is what really aligns with standard and guidance out there. But if your device is critical and contacts a sterile area of the body, then you're going to want at least two endpoints as well as those detergent residual test, such as cytotoxicity.
Okay, great. Speaking of cytotoxicity, this is how do I know if my device would require two endpoints for cleaning? Do all devices require two endpoints and cytotoxicity?
So No, not all devices require cytotoxicity. If your device is patient contacting and is going to contact a sterile area of the body or maybe mucous membranes, so semi-critical or critical classification, then I would recommend cytotoxicity. And same for the two endpoints to align with ST98. You're going to want two endpoints if it is semi-critical or critical. So once again, that's contacting non-intact skin or mucous membranes or entering sterile areas of the body.
Okay, great. We have time for maybe one or two more questions. Our next question is, does Nelson provide consulting to hospitals so that they can carefully and thoughtfully plan their timeline and budget needs to have the necessary testing conducted?
Yes, definitely. I can get you in touch with our consulting group, and they would happily work with you.
Great. Another question is here, back to cytotoxicity. Why is cytotoxicity performed on a single-use product after cleaning?
Yeah, so cytotoxicity would be something you want to consider for those, once again, patient contacting devices. And it is performed to assess and ensure that there really are no harmful residuals from the cleaning process remaining on the device prior to use on the patient.
Great. Well, we are about out of time for today. If you have additional questions, feel free, please, to reach out to Brianna, who I'm sure would be happy to address any of these questions after the event. This event will be made available on the Nelson Labs website within a couple of days, so that you can refer back to this presentation and also refer it to friends and colleagues as well. And on the screen, you also have Brianna's contact information, and that is how you would get in contact with Brianna. Again, we'd like to thank Brianna for this great presentation and for her insights on this. And we'd like to thank you, the viewer, for attending this session. We hope that you found it to be a valuable experience. And we hope that you have a great rest of your day.
Thank you.